How is everyone feeling after the exam? Good? Good? Excellent. Everyone feel all right about the exam? We're going to talk about it in just a minute. Before we get to the exam, I'll remind you again on Friday, probably Monday as well. But Monday is National DNA Day, in case you didn't know. I know, it's extremely exciting. In honor of National DNA Day, there are a bunch of things happening on campus, at the campus center. There's a crime lab, shark genetics, shark genetics, cancer center scouting, something going on, genetic counseling, all sorts of things going on at the campus center you should check out on Monday. I think there's a website up there somewhere. Yep. The website if you want to see what's going on. Hopefully everyone during this course has learned to love DNA. So you should check out DNA Day. Okay? Particularly if you're interested in molecular biology or some career in molecular biology, check out what's going on next Monday. All right. Yeah, shark genetics, Bailey. DNA Day. Check it out. All right. The exam. So thank your TIs once again. They spent 5 hours yesterday helping me grade. They were here until 10 p.m. last night. We got it all done. So you will get your exams back in recitation. Okay? I don't have an average for you because the TIs haven't entered the grades yet. But since we got them done this week, I'll give you until a week from Friday for point adjustment. So that'll give you a chance to get your exams back this week in recitation. Go over questions you have with your TI. If you have any additional questions that they can't address, then you can come and talk to me. As far as things, points the TIs can adjust, we did most of the adjusting last night when we were grading. So the only thing that they can fix for you is if there are errors in the addition of the points on your exam. Okay? So they can fix those for you. Anything about content and the way questions were graded, and if you want to ask about points, etc., that you have to come to me for. Okay? We did make one major adjustment on the exam that I want to let you know about. So does everyone remember this question? Yeah. How much you hated that question? Yes. All right, so as a class, generally, everyone bombed this question. Which tells me that you were confused about what it was that I was asking you. So what I was asking for was, tell me what's made and how that equals total ATP. So something like this. That's what I was asking for, which is not what most people interpreted that question to be. So I didn't feel good, given the fact that no one understood what I was asking for, giving that as a question on the exam. So I have taken that question off the exam. That means your exam total win is out of 113 points instead of 125. For the few of you who got this question right, so what we did for those of you who got it right, I turned those into extra credit points. So you got 1 extra credit point. There were 4, I think, parts to this question. For every one of those you got right, you got an extra credit point. So no one got penalized for it. If you got it right, you got a few extra credit points. Okay? But you should make sure in recitation this week that you go over this with your TI and then you understand what I was asking for. You know how I like to put questions on the next exam that everyone doesn't understand. So I suggest you figure this one out before the final. I'll try to be clearer if I ask something like this on the final. But you should definitely go through this and make sure that you understand what I was asking for. Even if I didn't ask it in a particularly clear way. Okay? Does that make sense to everyone? Overall, I feel like people did pretty well on this exam. You guys knew your pathways. Right? There weren't any surprises on, well, maybe except for this one. There weren't any surprises on the exam. So hopefully you will like how you did on your exam. But you'll get that back in recitation. All right, so are there any questions on the exam before we get started today? Any other questions? All right. And let's get started. So we are going to finish up photosynthesis today. Looking ahead, as of today, there are exactly 2 weeks of class left. With 2 more weeks of molecular biology to get through. So I think our last class day is on Wednesday. Our final, lucky us, is on the last day of finals, bright and early Friday morning. Friday the 13th of May. Yes. So, put that on your calendar. All right, everyone settle down. Just make sure you're paying attention to what's coming up. So we only have a few recitations left. Is that 2 recitations left, I think? And 2 weeks of lectures. In those 2 weeks, we're going to finish up photosynthesis today, and then we're going to get as far through signaling as we can before the end of the semester. So we're going to start with signaling in neurons and look at action potentials. And we're going to go into cell signaling and look at some of the signaling pathways that cells use. And if we get all the way through, we'll get to cell cycle and maybe a little bit about cancer. Okay, so that's what we're doing in the last 2 weeks. The final is cumulative. It is at least 25% of your grade, so you can't blow it off. You cannot drop the final. If you blew off one of your other exams, make sure that you understand that section. Right? It's all going to show up on the final. So start thinking ahead. All right, so we have to finish up photosynthesis before we get into signaling. This should look familiar. Showed it to you several times on Friday. We got most of the way through the light-dependent reactions. Those reactions in photosynthesis that require light as an energy source to drive those electrons into a higher energy state. Remember, for each of these linked pathways, you need to know what you're starting with and what you're ending with. Also how they're linked. Starting with the light and water, producing oxygen, ATP, and NADPH. Today we're going to move over to the Calvin cycle and see what happens there. Before we do that, let's remind ourselves of what the light reactions look like. There are 2 sets of linked pathways. Photosystem 2, which is picking up photons of light, transferring that energy to electrons, setting those electrons, passing them through a series of proteins to a cytochrome complex that is pumping protons across the membrane. Plasmocyanin is then passing that to photosystem 1, where again it's picking up photons of light, using that energy to excite electrons, passing it through a series of protein complexes, so finally those electrons end up in NADPH. This is also often called the Z scheme. Okay, so you tip it on its side, it looks like a Z. Straightforward. Energy, right? Energy of the electron picks up energy, loses it as it's passed from complex to complex, picks up more energy, loses it as it's passed on to the final electron acceptor. In our schematic, A little busier perhaps, but we show the flow of electrons, where protons are being pumped across the membrane. Remember that you need to keep track of what space these things are happening in. On the exam, some people got confused about the terms for spaces in the chloroplast for photosynthesis versus terms for the spaces in the mitochondria for cellular respiration, and the components of each of those. They're similar but slightly different, so make sure you know which goes with which. For the light reactions, we're in the chloroplast. We are in the thylakoid space. So inside those thylakoids, which are inside the double membrane of the chloroplast. Remember, you need to keep track of the flow of electrons, where they're coming from, where they're ending up, and then what's happening to that proton gradient that this moving the electrons around is creating. Another set of terms people got confused about on the exam that you want to make sure you get straight: photoreduction and photophosphorylation. Photoreduction specifically refers to the light-dependent synthesis of NADPH. We're reducing that molecule as the final electron acceptor. Photophosphorylation, the only thing getting phosphorylated in this set of reactions is that final ATP synthesis. All right, so photoreduction here, photophosphorylation here. So make sure that you keep those things straight. Let's do a few clicker questions to get everyone woken up. Use my clicker system. So let's remind you about the light reactions. Also keep straight where NADH versus NADPH is used. C for phosphate or for photosynthesis. Help you remember.
Oh, yours?
All right, everyone get their answer.
Oh, okay, right there. Like that. I see.
One more second. To get their system working. All right, let's take a look. All right, so we're getting those electrons in the light-dependent reactions from water. Let's talk a little bit about some alternate pathways for the light reactions. And we'll talk about, the last thing we'll talk about the light reactions are cyclic versus non-cyclic photophosphorylation. The photosphorylation, which molecule are we talking about? ATP. So we're talking specifically about generating that ATP. So if this is a scheme we've talked about, take a look at it for a second. Is it cyclic or non-cyclic? Alright. No one get their answers in? Let's get that flow of electrons.
Oh, yeah.
let's take a look. All right, a little bit of disagreement. Anybody want to justify their answer? Why non-cyclic? So there's no place where anything in that set of reactions loops back. Let's see here. All right, so it is non-cyclic. If we look at the flow of electrons that come from water that go into NADPH, there's nowhere where electrons or protons or any other molecule form a loop. So for this non-cyclic photophosphorylation, what does that mean for the input of water? If we use non-cyclic photophosphorylation to make ATP, how much water do we need? Everyone get their answers in? All right, let's take a look. All right. So if there's no loop anywhere, particularly for the electrons, we need a continuous source of electrons, which means we need a continuous input of water. Answer is yes. So non-cyclic, electrons go from water to NADPH. You need that constant source of electrons. So you need a continuous input of water. If we wanted to make this a cyclic cycle, where would you put the loop in? What is it that you think we want to cycle? What's moving through all of our protein complexes? The electrons. So we want to make a cycle with our electrons. Where would we put that loop in this system? Where does it make the most sense? We want to loop the electrons to the cytochrome. Why the cytochrome? So we can continue to pump protons across the membrane. What are the protons doing for us? We're going to use those to make ATP. So if we can keep protons pumped across the membrane, we don't need to keep getting new electrons if we form a loop. We put that loop before we stick those electrons into NADPH. We transfer them back to the cytochrome. Use those excited electrons from photosystem 1 back to cytochrome to pump more protons across, put it into photosystem 1, excite them, stick them back into cytochrome, pump more protons across. We can form a loop with those electrons that will continue to pump protons across the membrane, which will continue to give us a gradient, which will continue to drive ATP synthesis through our ATP synthase. All right, so it would look something like that. We're cutting photosystem 2 out of the picture, no longer needed. So in that situation, for cyclic photophosphorylation, what does that mean about the input of water?
No, just, oh yeah, get up and
electrons, which means we're not dumping them into ATPH, which means we don't need a continuous input of electrons, which means we don't need a continuous supply of water. So there are multiple pathways through these light reactions depending on the physiological state of the plant and what sort of environment it's in. All right. So just to keep you on your toes, during these light reactions, where is ATP made? What space? Oh, sorry. Try again. Sorry about that. All right, you need to know where the ATP synthase is and on what side of this ATP is made. All right, this is another mistake people made on the exam. For the electron transport chain, a number of people had ATP made on the wrong side of the membrane. All right, which side of the membrane you're on is important.
Because the membrane is an inner part of the matrix.
I think it's
Yeah, that's what I said. No, it's the product comes out. It's whatever the outside. Yeah, but that comes with the middle, right? Isn't it inner and then
Do I got it figured out? Let's take a look. All right. So the correct answer is the stroma. Why not the matrix? Because that's mitochondria.
the wrong thing.
The wrong thing. Right, so just to get back to our original drawing. Remember this all happens in the thylakoid membrane. We're pumping protons into the thylakoid. They're rushing back out into the stroma, and that's where the ATP is made. When you're thinking about the movement of protons, remember the whole goal of this is to set up a proton gradient. It's easier to set up a high concentration of something in a smaller volume. So if you notice between electron transport chain and our light reactions, the protons are always being pumped into the smaller space, into the thylakoid space or into the intermembrane space. Right? Smaller space, easier to build up that concentration gradient. And then they're coming back across into either the stroma or the matrix, depending on what set of reactions you're talking about. All right, so you need to keep track of what space things are happening in and what direction they're moving across the membrane. So if we go back and look at our scheme again, our non-cyclic light reactions, if we wanted to make this cyclic, we'd send our electrons from our ferredoxin back to cytochrome. Right? That's got a higher energy than cytochrome does. So it can pass it back to cytochrome, pump protons, and then send those electrons to photosystem 1. Getting rid of that, all of those components of the non-cyclic pathway. And with no additional electron input, a chloroplast can continue to pump protons across that membrane and continue to make ATP. All right. That is it for the light reactions. So we've talked about all these light reactions. They happen in the thylakoid membranes, moving things back and forth across the membranes from the thylakoid space to the stroma. They're linked with the Calvin cycle through those energy molecules and electron carriers that are made, so the ATP and NADPH. So they're made in the stroma. The Calvin cycle, which is the cycle that's going to produce our carbohydrates. So the sugars that are being made from all of this are made in the stroma. Right, so that energy and electron carriers are made in the stroma where they can then be immediately used for the Calvin cycle. So let's look at this half of the photosynthetic process. Remember, you need to keep track of what's going in and what's coming out. You need to know the substrates that are used and the products that are made, both the light reactions and the Calvin cycle. So in the Calvin cycle, we are using ATP and NADPH as energy. And we're using carbon dioxide to make organic molecules. So those carbohydrates, those organic molecules that are made, those are our products. And this all occurs in the stroma. So we're going to take a look at the Calvin cycle. We're not necessarily going to look at every single step in that cycle. We're going to look at the important phases and what happens in each phase. There are just a few molecules you're going to need to be familiar with. One of them you should already know, so it'll be review. We're starting off in the Calvin cycle with our carbon dioxide and a 5-carbon molecule called ribulose 1,5-bisphosphate. The enzyme driving this cycle, that's going to make that initial step of adding the carbon dioxide to this ribulose 1,5-bisphosphate, is ribulose 1,5-bisphosphate carboxylase oxygenase. Hard to say. So that has been shortened to Rubisco. That's much easier to say. That Rubisco adds that carbon dioxide to that 5-carbon compound. Rubisco is found in all photosynthetic organisms that use this Calvin cycle for carbon fixation. So the step of adding the carbon dioxide is carbon fixation. And it's thought that it's the most abundant enzyme on the planet is Rubisco. It's also an extremely slow enzyme, on the order of 3 reactions per second. Pretty slow for an enzymatic reaction. So adding that carbon dioxide to a molecule takes some time. All right, so that ribulose bisphosphate carboxylase oxygenase, or Rubisco, takes that carbon dioxide, adds it to our ribulose 1,5-bisphosphate to form this intermediate. So now we have 6-carbon molecule intermediate. That's you. And then splits it apart into 2 3-carbon molecules. So it's starting to seem a little bit familiar, maybe. These 3 carbon molecules are 3-phosphate glycerates. And this is the step that starts the cycle. So we know that it's a cycle, and we know we're putting ATP and NADPH in. So you need to keep track of where we start and end, same place since it's a cycle. We're going from a 5 carbon molecule plus a carbon dioxide through visco, ending up with 2 3 carbon molecules. We put in some energy and electron carriers, and we end up with something that comes out of that cycle. And what remains then is regenerated back to our 5 carbon molecule. You might notice the numbers don't really work out here, right? They don't add up. So we need to look a little more closely at each step. So the cycle doesn't deal with sort of just one molecule at a time. To make this work, we have to start with 3 5 carbons, the ribulose 1,5 bisphosphate plus 3 CO2. Those together make that 3 of those 6 carbon intermediates, and are split into 6 3 carbon molecules. We add a phosphate, right? So if we're adding ATP, we're breaking that bond. Where is that phosphate probably going? Onto the substrate. So we're not looking specifically at those molecules, but we're adding phosphate, rearranging. Right? Here we're rearranging. And these steps are simply to get that molecule that we're starting with to glyceraldehyde 3-phosphate. Where else have we seen G3P? Glycolysis. So this is a molecule that the cell knows how to deal with. It's already got pathways in place to do things with G3P. So notice that, right, 6 3-carbon molecules that we undergo a series of rearrangements, phosphorylation and then rearrangement. The cycle spits out for every turn 1 G3P. The remaining 5 3-carbon molecules are used to regenerate back to our ribulose 1,5 bisphosphate. So notice in this next step, we have 5 3-carbon molecules, 15 carbons total. They're rearranged into 3 5-carbon molecules. And then a phosphate is added, and we're back to ribulose 1,5 bisphosphate. So we can break that down more specifically into 3 phases. The carbon fixation phase, right, where we go from 3 5 carbons plus 3 CO2 to 6 3 carbons. So that's phase 1. So we're simply adding the carbon dioxide. Phase 2 is the reduction phase. So we're adding a phosphate and then stripping it back off to get the proper rearrangement to end up with 6 glyceraldehyde 3-phosphates. One of which gets kicked out of the cycle. The remaining 5 G3Ps enter the regeneration phase of the cycle, where those 5 G3Ps get rearranged into 3 5-carbon molecules, a single phosphate. We add in 3 ATP to get that additional phosphate back on there and end up back at ribulose 1,5 bisphosphate. So that's the full cycle. We need to keep track of the what's happening in the 3 phases. When you get a shift in the carbon molecules, so what size carbon molecules are you dealing with? And where is energy being put into the system and what's coming out of it? So where CO2 is going in, where your energy and electron carriers are being used, and what's coming out of that cycle. So to put it in sort of a stepwise picture, 3 ribulose bisphosphate plus 3 CO2 produce 6 3-phosphoglycerates, or 3 carbons. Those 6,3-phosphoglycerate plus 6 ATP plus 6 NADPH produce 6 G3P. One of those leaves the cycle to make sugar. The other 5 are used with 3 additional ATP to regenerate our 3 ribulose bisphosphate. All right. And in this step, ATP phosphorylate our molecule, and NADPH reduce it to get to that G3P. All right, another view of it. What happens to our G3P that comes out of the Calvin cycle? What's one thing that we know a cell can do with G3P? Makes pyruvate. It can make it into, turn it into pyruvate by going through the rest of glycolysis. And we know what happens to pyruvate. Where does pyruvate go? Goes in the TCA cycle in the mitochondria. Cellular respiration makes ATP. So that's one thing that can happen to these G3P. 2 additional pathways are from the stroma and the chloroplast. The G3P produced from the Calvin cycle can stay in the chloroplast, in the stroma. Remember that G3P and DHAP interchangeable, flip back and forth. So you always have some component of DHAP. But inside the stroma, or it can be sent back out into the cytosol. And the cytosol can be turned to pyruvate and sent to the mitochondria. Or it can go through a similar pathway in either the stroma or the cytosol. You take a look, it looks pretty similar to glycolysis in reverse. That first phase, so we're going from G3P to fructose 6-phosphate. There's 2 of these to make a 6-carbon molecule, rearranging our fructose 6-phosphate into glucose 6-phosphate, which is where we started with glycolysis. There are a few more rearrangements past that. So in the cytosol, G3P can be taken to glucose, added, combined with a molecule of fructose to make sucrose. So what's the difference between glucose and sucrose? Sucrose has how many simple sugars? The disaccharide. So sucrose is a disaccharide, the molecule containing both a fructose and a glucose together, whereas glucose is just a single simple sugar. So in the cytosol, G3P can be taken through to glucose, and then with the fructose 6-phosphate, which we've already made earlier in this pathway, can end up as sucrose and passed around for energy. If it stays in the stroma, it can go through the same pathway, but it ends up in glucose and can be turned into starch. Starch is a polymer of glucose molecules, so lots of glucose molecules together in a chain. So in the cytosol, the 3 different things that can happen to that G3P that's kicked out of the Calvin cycle. Turn to pyruvate, will sent to the cytosol and either turn to pyruvate for ATP synthesis in pathways we've already talked about, or converted to sucrose and then sent throughout the plant as the energy molecule. If it stays in the stroma, it can be turned into starch, that polymer of glucose, many glucose together in a chain, and it can be used for metabolism when there isn't any light, when there isn't any light to drive those light reactions. All right, so that is photosynthesis. We've talked about the light reactions. We've talked about the Calvin cycle, what goes into each, what comes out of each, how they're linked through those energy molecules and electrons. they use water, they produce ATP, they produce NADPH, they produce oxygen. In contrast, the Calvin cycle uses that ATP, uses that NADPH, uses carbon dioxide, and produces our carbohydrates. All right. That is photosynthesis. Any questions on photosynthesis?
We know the names of all the molecules, like we did for the TCA cycle.
You mean for the Calvin cycle?
Yeah.
You do not need to know the names of all the molecules other than the ones I've mentioned in class. So you need to know ribulose 1,5-bisphosphate. You need to know Rubisco, or at least what Rubisco is. You need to know the 3-carbon compounds, the phospho-3-glyceraldehyde, and then the G3P, which you already know. And then just what happens in each phase of the cycle. Okay? Where you get changes in your carbon molecules in terms of the number of carbons they contain, where ATP is used, where NADPH is used. But I don't expect you to know every single step. There are a lot of steps in there that are conversions of molecules. Any other questions about photosynthesis? All right, for the last few minutes of class, I just want to introduce you to what we're going to talk about on Friday, because we're sort of shifting now from energy and metabolism to signaling. So a quick introduction to signaling to get you started thinking about what we're going to talk about next. So for the rest of the semester, we're going to be talking about signal transduction. We're going to start off with neuronal signaling. We're going to look at action potentials. But let's think about signal transduction more generally. So in a biological system, if a signal is sent out, right, that biological system has to be able to detect the signal. It has to turn that signal into some sort of information a cell can use. And then there has to be a cellular response to that signal. So think about these three things as we go through these signal transduction pathways. What's the signal? How is it detected? How is it converted to information? What change in the cell does it induce? We can think of this just to give you a framework of signal and signal detection. We think about how people communicate, what the signals we use are, how we detect them. Right? And specifically, both in the terms of communication between people and communication between cells, what's the role of distance and how you're communicating? Right? So we have some friends who would like to communicate with each other. Right? If you are sitting next to your friend, how would you communicate with them? Right? You might be able to punch them in the arm, poke them if they're sleeping. Right? So there's direct contact, shake hands. Right? If you are very close to the thing you're trying to communicate with, the person you're trying to communicate with, you can communicate through some sort of interaction, physical interaction. All right, what about if you wanted to communicate a friend who's a couple of rows away from you?
Hi.
I can.
Right? If I wanted to say something to Sai, I would speak to him. Right? And so the signal would be my voice, the detection would be his hearing system, we would be able to communicate. So I'd have to amplify my signal. I might have to add in some of this, do some interpretive dance to get your attention. All right, so we've added both a signal and a mode of detection. All right, so now you have to detect my signal. Two modes, you could hear it or you could see it. What about if you're on the other side of an ocean, right? We're on an island, we know lots of people other places are not on this island. There's an ocean in between. You can't yell at them. We can't touch them. You could Skype them. Let's imagine the internet doesn't exist.
Carrier pigeons.
You have to send some signal, right? Carrier pigeons. Back in the days when people actually wrote letters. Right? Skyping even. That's not a direct interaction, even though it feels like it. Sending electrons back and forth. Alright, so the signal changes depending on the distance between the two things that are trying to communicate. So think of that in terms of cells. And we'll do this first thing on Friday. I'll put this up for you to look at in the lecture. We're going to match the signal mode in cells with the distance over which it acts. Start there for signal transduction.
Okay, I'm ready to